Good morning, everyone. Good morning. This is a wonderful session today from the Teacher Institute for Evolutionary Science TIES. We called it TIES webinar series. My name is Bertha Vasquez, and I'm the director of this wonderful teacher-run network, and I'm also a science teacher in Miami-Dade County. To tell you a little bit about TIES, we're a group of about 70 teachers nationwide right now that present workshops on evolution to their fellow science teachers around the country. A special shout out to the teachers joining us today from Arizona, courtesy of the Arizona Science Teachers Association, and to teachers in Bay District schools in the Panhandle of Florida and Hillsborough County schools, which is around Tampa, Florida. And today we have a great presenter and I think friend at this point. I'd like to say Dr. Reimers is a computational biologist at Michigan State University. He's been a bioinformatician at the Karolinska Institute in Stockholm, at NIH, and at Virginia Commonwealth University. He's reviewed the literature on SARS-CoV-2 evolution as he is preparing a COVID-19 research proposal. I think this is a very timely session today, especially here in Miami-Dade County. And I want to share a t-shirt with you before we begin. Yes, that's why we're all here. We want to learn and we want to aspire to be a little less wrong. And Dr. Mark Reimers is going to help us do that today. Good morning, Mark.
Good morning, Bertha. Thank you for hosting me and arranging this. And Bertha, if you would help monitor the chat, and maybe I can pause for questions every few slides. I'd like to sort of engage with where people are asking.
Yes, as a matter of fact, you already have a question, so that would be a good idea.
Okay, well, let me get started and then we'll pause for questions in a little bit. And now I'm trying to find where that share screen is again. Here we are. Good, okay. And we'll share screen one. Okay, can you all see this?
I can.
Okay, great. So I'm going to talk about how the coronavirus evolved, how it is evolving now, and what impact it might have on our own evolution right at the end. But let's start with just a question about, well, what is a coronavirus? I think you've probably seen too many pictures of coronaviruses by now.
Yeah, indeed.
As of early 2020, there were 40 known types of coronavirus. There are many more known now, we know that they're out there, even though we don't know much about them. And they got their name from the very early electron microscope pictures. They were made in the 1960s, and here's one of them on the bottom right, where when you take a section of them, you can see all these sort of spike proteins along the outside, and people thought those looked like the points of a crown. So that's how they got their name of a crown virus, or using the Latin term, a coronavirus. Now in 3 dimensions, we now know that they look more like a naval mine, the old style World War II naval mines. And this is the famous picture that's become quite widely distributed. And this is showing the spike proteins in red, and those are the proteins that sort of grab hold of a receptor on human cells. This is an RNA virus, so there are many kinds of viruses. There seem to have been many separate evolutionary or origins of viruses, so they may have been, whereas all multicellular life comes from one origin, it seems that viruses come from several different origins. So there are DNA viruses and RNA viruses and several different kinds of RNA viruses that have nothing to do with one another. And this is a so-called positive strand RNA virus, so it's a little bit unusual. What's really unusual is it has such a long genome. RNA viruses typically have genomes that are about 10,000 bases. This virus has a genome that's over 30,000 bases. It's very, very long. And what does it do? Well, how does it infect us? we know of 7 coronaviruses that infect human beings, and 4 of them, you've probably already had an infection by one of these coronaviruses. So 4 of them, and I've indicated the names in the second line up here, 4 of them give you a sort of mild case of the sniffles. Most of us have had one or more of these. And then there are 3 coronaviruses that are known to cause very serious diseases, SARS, MERS, and COVID-19. And COVID-19 is caused by the SARS coronavirus 2. So coronavirus disease 19 is the disease caused by the coronavirus SARS-CoV-2. And it infests cells in the lungs, also in the gut, and also it has the bad habit of interfering with immune cells. So in that way, it's a little bit like the AIDS virus, which is a completely different kingdom of viruses, but it acts a little bit like that. And it turns out that actually most, not all, but most of the deaths are actually due to immune overreaction, meaning that your body has a hard time clearing it, and then eventually pulls out all the stops, sounds all the alarms, and all of your immune system gets called to ready. And that overreaction is what brings on most of the deaths from SARS-CoV-2. And here's a picture of roughly the virus's life cycle. It enters through this protein called ACE2 with the aid of this other protein, TMPRSS2, which then cleaves the spike protein, allows the virus inside the cell, and then it opens up and starts replicating. So let me just pause for a minute here and ask if anyone wants to ask a question or comment on anything so far.
We have several questions. This one's from Alan John Gray.
Okay, Bertha, I can't quite hear you, so you may need to speak up.
Haha, how's that?
That's
Is
better.
that better? I had my mic up.
Sure.
Alan John Gray says only a few viruses are enveloped with lipoprotein, presumably from the host cell plasma membrane. Have the coronaviridae acquired this envelope, or has it been retained from an earlier common ancestor with a non-enveloped family? In either case,
Okay.
What are the evolutionary advantages of having the envelope?
Okay, so I can't really give you a good answer to the second question, but I can answer the first. So all of the known coronaviruses do have these envelopes, and we believe that this is an ancestral feature. They belong to a broader family that shares this trait, but we don't really know where it originated. So the evolutionary history of viruses is very difficult to trace because they mix and match genes so readily, and they even pick up genes from their hosts. And so it's very difficult to know exactly where they came from and where in evolutionary history this particular lipoprotein envelope came from. But all of the known coronavirus, it's a distinctive feature of all the coronaviruses and their relatives.
Next question from Kate. Have we isolated the vector species? The most recent update I heard was the fruit bat.
Well, we know that they originally came from a species of bat in China. And I will be talking more about that in just a minute. Okay. So if you can hold that question for just a minute.
And the other question is where I got my Aspire to Be Less Wrong shirt. So I'm going to go ahead and put that, the link in the, I'm not making any money here. This is not a TIES project, but there's the link. I bought them for TIES teachers for future workshops. So, okay.
Great.
I think
okay,
we're ready
let's
to
go back then. Going back to the presentation. So let's go back, I think, because here, yeah. Okay, so let's talk about the evolution of the coronavirus. So most of the known coronaviruses infect bats and birds, and we have good evidence that there are many other coronaviruses that infect bats and birds that we have not identified yet, but there seem to be lots more. Now, partly this is an answer to the question that just came up. So all of them seem to have a very similar kind of genome. They're all about the same length. Some are a little longer, some are a bit shorter, but they all have the same basic structure that is shown here on the bottom right. So there's this set of two long genes which then get chopped up. They get translated and transcribed and translated first, and those make all the other proteins that are needed for the virus's life cycle. And then these are the proteins that are involved in the coat, in particular the spike protein, which is indicated here in red. And you have the same common structure among a variety of human coronaviruses and the coronaviruses that infect other animals. Now, we can then compare these genomes and look for the discrepancies. Most of these are, 85, 90% identical. Some of them are, 95, 97, 98% identical. We can trace their evolution by looking at where the changes have occurred. And this picture here on the left is from an article near the beginning of the year. Now, if there's so many more coronaviruses known that it'd be impossible to read. Well, it's pretty difficult to read that right now. But you can see that that there's a number of different subfamilies of coronavirus. The Wuhan coronavirus 2 is in red here, and it seems to be related to a number of bat coronaviruses and also to the SARS coronavirus. There are other different families of coronaviruses. You can see that most of them infect bats. Some of them also infect domestic animals. So up here is, let's say, the bovine respiratory coronavirus, a rat coronavirus. So they have gotten into other animals. Now, one of the big controversies is, where did they all start? So when, this is your divergence point. When did this family tree explode? We're used to looking at evolutionary diagrams where the origin is, millions or hundreds of millions of years in the past. And the way we get those origin times is by looking at the sort of rates of genome evolution as far as we can estimate those, and then sort of extrapolate backwards and say, well, if there are, 300 base changes from here to here, then, or 500 base changes, then how long would that take to happen, given the, typical rate of base changes in these things, which is, a couple per year. So the current estimates, most of the current estimates are sometime within the past 10,000 years, and the best date we have is about 8,000 years ago for this. And there's some argument about this. Let's be fair. Some scientists say this just seems far too short. How can you have so many distinct coronaviruses that infect bats in different parts of the world and birds in different parts of the world? How can they all evolve so quickly? But the evidence that we're used to, that we've used on most other evolutionary reconstructions does suggest a very recent explosion of these coronaviruses. And the standard answer is, well, birds fly and bats fly, and they can fly very long distances. So let's just say that this is not for certain. It is always somewhat chancy. We don't know that evolution proceeds at the same rate. We usually assume so, and often it seems to be right, but sometimes that's not true. But if true, it's a very surprising finding that this whole group of viruses seems to have spread and proliferated so recently. And it makes you wonder, well, how do these things jump species? How do they find new targets? Well, they find new targets by being in close proximity with animals. And where bats usually hide out in places that we don't like to go. But so when have we started to be in contact with lots of bats? Well, as we expanded and as we started clearing a lot of land for agriculture, as our species cleared a lot of land for agriculture, we have been, tearing down a lot of bat habitat and putting our domestic animals in places where bats live, where they wouldn't have met them before. So my opinion, and this is just an opinion, is that it's partly, if not largely, due to the human impact on natural ecosystems that have brought so many new sort of customers to the coronaviruses, so many new potential targets. So why do bats and birds seem like good hosts for not only coronaviruses, but lots of other viruses? Of course, Ebola and Nipah and many other viruses are spread by bats. Well, part of the reason is the way that bats and birds have evolved their own immune systems. So they're certainly subject to lots of microbial attacks because they roost in crowded conditions. So infections spread through bat colonies in, very, very quickly, in months, because they're exchanging microbes so readily. But whereas most animals, like ourselves, deal with an infection by developing a fever, and that because most viruses and many bacteria don't reproduce as well at temperatures slightly higher than normal, bats and birds don't. And part of the reason is that they are already flying a large part of the day. And so that flight, they're burning a lot of energy. So their body temperature rises. So they kind of have a natural fever already. So they don't need to be the sort of typical fever that we have. The other thing is that they're really energy limited. They're really chowing down a large part of their body weight every day in food because flight is very, very demanding. They're exercising a lot, so they're eating huge meals, and they, in some sense, don't have the energy to spare. The fever does take up a lot of energy. So their immune systems have evolved in a different way than ours so that they damp down those energy-intensive responses like inflammation or the cytokine storms, where our immune systems signal that, all the alert. So they respond to infection with very slow, more targeted antiviral responses like our adaptive immune system, but they don't have the inflammation or fever that most mammals have. And the result of this is that bats take a long time to recover from infections. They sustain low-grade infections, but they don't feel them. They don't have the inflammation and the secondary symptoms from a lot of these infections. So let me come to the question that was just asked. What was the intermediate species? Well, it's clear that the coronavirus 2 came from bats in the relatively recent past, but it does seem to have adapted some. It's not this coronavirus is not identical to any known bat coronaviruses. So one common route is that the coronaviruses infect domestic animals, which they have more contact with, and then humans. So several of our common cold coronaviruses first infected cows and then came to us. And we know that because we can look at the genomes and see that our version looks more like the cow version than like the original bat version. So we still don't exactly know, but the consensus is it's most likely that the SARS coronavirus 2 may have crossed over via pangolins, but pangolins aren't domestic animals. And it's not entirely clear how they did that. So we've now isolated coronaviruses in pangolins, and they are not exactly the same. So it's still somewhat controversial, but it seems like that pangolins were most likely involved. And before I get to some of that, let me just say that the current most likely place for this happening, the Wuhan live animal market, is a place where, you know, the carcasses of animals and human beings eating lunch are all crowded together. There's viruses all around. Most of these viruses won't, you know, find a suitable habitat in human noses and lungs, but all it really takes is one. And so far as we can tell, all of the SARS coronavirus 2 genomes that have been sequenced, and thousands of them have been sequenced, they seem to originate from a common ancestor in late fall, perhaps November of 2019. So, you know, it seems to have happened just once, and all of the coronavirus infections that have swept the globe seem to be traceable to that one infection where in Wuhan. So now we come to the question of, okay, what made it possible? So, you know, a virus needs to grab hold of a protein on a cell surface. And here we have a model here of the spike protein grabbing hold of a human angiotensin converting enzyme protein, which is a cell surface protein. And that's because of a mutation in the spike protein, where it seems like what's happened is the bat version of the spike protein was substituted with a pangolin version of the spike protein. And that pangolin version seems to be a lot more suitable for entering human cells than the bat version was. And that seems to be what sort of ramped it up by, ramped up its infectivity by several orders of magnitude. Another thing that happened was that there was an insertion of a number of amino acids that are basic that enables that spike protein to be cleaved more readily, so that when it's attached, then it splits open and then lets the coronavirus enter the cells. And again, that's something that we don't see in the pangolin version, we don't see in any of the bat versions, but it's the kind of insertion that could happen. Again, a rare event, but something that could happen and does happen in viral evolution. It's been demonstrated that it's the sort of thing that could easily happen to a spike protein. So let me just finish by saying that, you've all heard the conspiracy theories. I think it's not absolutely, batshit crazy that somebody could make a virus, but there's no evidence that that this particular virus was synthesized, and attempts to do things like this have not been terribly successful. We know that there have been some attempts that have not worked. But of course, it's something that could happen. Biotechnology is getting better. The more serious sort of conspiracy theory is that, well, what about this virology lab that's not too far from the seafood market? They've cultivated a lot of bat coronaviruses, and one of them is relatively similar to the SARS-CoV-2. Well, again, it's not inconceivable that something like that could have escaped. In fact, we know that viruses occasionally do escape from these high containment labs. However, the SARS-CoV-2 sequence diverges at more than 300 bases from the closest sample there. And again, you can imagine, somebody very patiently over many years tinkering with this, virus to, at 300 bases. But to do this by hand seems a lot of work, and there's no evidence that that was done. So it seems that there's been a lot more evolution. It doesn't seem likely that this is something that was an escape. So let me stop sharing and come back and then take any questions.
Okay. I'm waiting on questions. Actually, you don't have any new questions at this point.
Okay.
Does
anyone want to ask a quick question, or then I'll if not, I'll finish.
Don't see anything.
Okay, well, let's go back, and then I'll just talk about sort of how it's evolving now. And then we can take some more time for questions. Okay, so I'm screen sharing, and it should be back, okay? All right, so how is it evolving now? There have been a number of mutations, a few dozen mutations that have occurred and have been copied. This is a diagram from a recent paper just tracing all of the different genotypes that have been sequenced of the coronavirus. So you can see that there's close to 100 known, but each branch point refers to one mutation. And the colors refer to where the mutation occurred, or at least where that sample was found. And it sort of gives you information about the spread of the coronavirus. So the origin, the original virus, of course, started in China and has largely been found in China and East Asia, but there are several distinct variants that have spread to Europe and several distinct variants that have spread to America, some of which came directly from China and some of which seem to have come via Europe. And now, of course, there's many others that are spreading more widely. Now, we've had now millions of infections, over 10 million infections worldwide, and yet I'm saying there's really only a few dozen mutations that have occurred, and that's because coronaviruses evolve very slowly. If you had the same number of infections of the flu virus, there would be so many dots and branch points on this page that it would be just unreadable and completely meaningless. So most of the RNA viruses, like the influenza virus, evolve fairly rapidly, and there will be hundreds of mutations every year. But the coronaviruses seem to evolve about at a tenth the rate that the flu viruses evolve, or maybe even a little slower. And that's because of these proofreading proteins that are in the first part of their genome. So far, there's relatively little evidence of evolutionary change. I should have just said evolutionary change, meaning that the coronavirus is moving in a systematic direction. So what you see here is, from the original virus, there's all kinds of mutations that are around, but there doesn't seem to be a trend. It doesn't seem to be going in any particular direction. Maybe around here, there seemed to be more proliferation. So there's one mutation that did occur here and has been spreading, particularly in North America, and that seems to make it a little more infectious in tissue culture. It seems to enter cells more readily, but it does not seem to make it more harmful. The general evolutionary theory will predict that disease microbes, once they make a jump to a new species, they will evolve to be, to spread themselves better, so to be more transmissible. So that'll mean they will make it more likely that people will spread them by sneezing or more gross means. But the virus is attempting to get more copies of itself out into the world, where other people can pick them up. And sometimes that means that they incapacitate the carriers less, meaning that they don't, if you're feeling so awful that you're lying down, you're not likely to spread the virus as well. Now, SARS-CoV-2 is in some sense is already in a sweet spot. It's very transmissible. And the reason it's very transmissible is because most carriers aren't knocked out. Most carriers have only mild symptoms or even none at all. And so they can spread the virus quite readily. We think that in some sense SARS-CoV-2 has already found the sort of sweet spot where where some people are very, very ill, but most people are only slightly ill. Now, one thing you might ask is, turning this the other way around, SARS-CoV-2 is evolving, but will it affect our evolution? So, any kind of plague might well affect human evolution, but we think that SARS-CoV-2 will have little effect on the evolutionary direction of human beings, because it doesn't seem to affect people before they reproduce or during the time that they're parents. It mostly kills or harms older people. However, there certainly are some genes that would be selected against if it did have a strong selective pressure. For example, type A blood. So I have type A blood, so I have about a 50% higher risk for getting severe COVID-19 than someone with type O blood. There's also a genetic variant. We think it's we don't know which gene it's in yet, because there's a whole group of gene variants that are tightly linked, that are inherited together. And we think that the version of those genes that comes from the Neanderthal ancestry increases risk almost by a factor of 2, almost doubles risk. And this, interestingly, this Neanderthal variant occurs more commonly in South Asia than in Europe. Most Neanderthal genes are more common in Europe. However, it's certainly the case that that epidemics can affect human evolution, and this is something that we live with most of the time. So we certainly see evidence that things like the Black Death, which killed almost half the people in Europe, did very strongly select for certain variants in the immune system, and many of them are the so-called major histocompatibility complex, or MHC, region on chromosome 6. And one of the side effects of this rapid and severe evolution is that our immune systems have been tuned up to be more responsive, to be quicker on the trigger, to mount a very severe inflammatory response at the slightest provocation, because of all of these plagues like the Black Death in the past 10,000 years. So we have evolved our immune system to be more on alert, but that means that when we're not getting infected, that readiness for inflammation is now a major factor in many of the chronic diseases like human heart disease. Interestingly enough, our close primate relatives don't get the kind of heart disease that we get. And they don't get a lot of autoimmune diseases, and they don't get a lot of other kinds of chronic diseases that human beings, that many of your parents or grandparents are living with and suffering from now. And it seems that it's largely our history of plagues, which has been partly because of our expansion into the natural world, that we are at higher risk for these things now. So that's where I'm going to end the meditation for today, and I'll take any further questions. Looks like lots of questions now.
Yeah, lots came in. Well, one of them you answered. They asked, Tracy Denny asked, what are your thoughts about how it will evolve in the future? You answered that, but it's tying into another question. Where is it? I think it's here, because people are putting questions. Yes. In the various mutations, what is the significance of the length of the line? The thing you showed with the...
Oh, yeah, that was basically saying how unlikely, or how distinctive that mutation was.
Okay. Well, thank Carly Karras for that question. You kind of also answered Kate Jones' question. What are the chances that humans will become symbiotic with COVID-19 as we are friendly with all the other microbes in our bodies, could it be useful to us instead of killing us? Actually, you talked about it becoming less harmful in some people, so you didn't really answer
Well,
that
if
question.
I can address that, I don't think it's ever going to be friendly. I mean, there are microbes we can tolerate. There are microbes that we depend on in our guts. I don't think any respiratory microbes or viruses are friendly to us. They're all, ranging from we can tolerate them to them being very harmful. And it may be that this coronavirus will, become like the common cold coronaviruses, that it will
Right.
evolve to be relatively mild, or it may not. But I think we will be. I don't think it'll ever be a friend.
Okay.
I think it's going to be. Hopefully, the best we can hope for is it becomes less harmful.
Right.
Uh, but I think the most practical thing to do is to get a good vaccine or a good treatment. And that's because evolution is going to take a long time. I don't think we want to wait for it to
No.
to evolve on its own.
Doug Bender has a question that I've read some of the answers to. He asked, "Can you explain the dropping death rates?" And I know that we did learn a few things in New York about how to position a patient in serious condition. Also, some of the drugs are working on the serious end of the
So I
scale.
can
But
just tell you what I've read from the medical people. I'm not a practicing physician. So the medical people say that the changes they've learned to sort of put people over. They're less shocked, so that by it, so they know better what to do. They know that they want to avoid intubating people if at all possible. So the recovery rates are better from the severe coronavirus. The other thing is that a lot of the infections in the United States in the past couple of months have been among younger people. Whereas the early infections, these were the severe infections. We knew about them because people were coming into the hospitals in New York and in Detroit, and they were, older people who were very, very ill. And before the retirement homes really strictly enforced distancing and masks, the coronavirus spread very, very rapidly. You may have noticed that old people tend to spit a lot more saliva out when they talk. One of the many indignities of old age, so it just spread like wildfire in those places, and of course people were so vulnerable. So it's better precautions on the part of the older people who are more vulnerable, and the perhaps a more cavalier attitude among the younger people who are not likely to get as severely infected.
right.
So those are, I think, the things.
We have a couple questions about children. Mark Coop is asking, younger people have been getting Kawasaki syndrome, where it does not show in older people. Why is there a difference? And Felicia Smith is asking that the younger people seem to have rashes. And
So that's
why?
beyond my area of expertise. Of course, I've read what you've read about Kawasaki syndrome and how it, there seem to be a small number of kids who are getting this Kawasaki syndrome, and they all seem to have antibodies to the coronavirus. But why that happens, I don't know.
I want to add that someone is commenting, this was a superb presentation. I read Nature and I read Nature and Science every week and had not heard such a nice synthesis. Leave it to a scientist doing bioinformatics.
Well, thank you. Thank you for your kind words.
So why are experts hedging on us gaining long-term immunity as we do with other viral diseases?
Part of the issue is that the 4 known coronaviruses, which have been infecting people, do raise antibodies, and that's how you get over the infection. But you can get reinfected with those. So the common cold viruses, of course, you know that you can get colds again and again. You get a couple of colds every winter, at least in the northern parts of the country. Usually they're different viruses. Many of them are rhinoviruses, but these coronaviruses you can catch several times during a lifetime. So we think that it may not be so easy. Now, part of the debate is about, well, if you get a serious case of coronavirus, like a serious case of anything, your antibodies seem to be stronger. So the one study that I read showed that people who had a mild case of coronavirus, of COVID-19, their antibody titer went down over the next 2 months. Whereas people who had a severe case, their antibody titer was staying pretty strong. So that's not good news, because of course what we would like is, for the vaccine, which hopefully will not give us a severe case, we'd like for the vaccine to protect us, but it's somewhat unclear whether it will.
Yeah, I've been seeing people, their antibodies disappear over time, which is very scary. We have a question. Do you think the food habits of humans is playing any role in controlling the COVID-19?
Well, I would say it's not playing a role in controlling, because I think our food habits brought it to us. I think if we want to, in my opinion, I think it's more than an opinion. I think nearly every microbiologist that I know shares this fervent belief that we are sitting on 2 time bombs. One is the rise of antibiotic resistance because of indiscriminate use of antibiotics. So they are shaping the evolution of hundreds of different disease microbes in a way that's unpleasant for us. And second, by cutting down the last remaining habitats, we are exposing ourselves to all kinds of viruses we've never encountered before, which are going to be very deadly. I mean, Ebola is, I think, one of the worst possibilities, and after it was first noted in a small valley in the Congo, it didn't appear again for 10 years, and now it seems to be appearing every year. So those are the 2 time bombs we're sitting on, and I think our food habits have a lot to do with the second, or our agricultural practices, and arguably have a lot to do with the first. So I think in both cases we need to really rethink how we live. So those are our big questions.
Are humans with autoimmune diseases more or less susceptible to the virus?
That's a very good question. I don't actually know the research on this. So, I don't know the evidence, but it sounds like the sort of thing that I would expect. I would expect that people with an overactive immune system would be more likely. But of course, overactive immune system there's several different ways your immune system can be overactive. And I suspect that only one of them would actually be protective. So probably there's some research on that. I just don't know what the answer is. But that's
how risky is it for someone, say, with COPD to use the antibody plasma to help with immunity?
That's really a medical question that's beyond my expertise. I couldn't say that.
And is the antibody response and immunity difference an IgE, IgG issue?
Well, there's many different immunoglobulins. You mentioned IgE and IgG. So the antibodies are going to be IgG, so they're the level of inflammation is partly tied to IgE, but partly tied to other things as well. So it's it's related, but there's much more. They don't work completely separately.
Okay, and I've got 2 more questions here. This one's from Nick Kaiser. So the difference in disease severity that we're seeing asymptomatic versus symptomatic, for example? Does it have to do with mutations within the virus itself, or does it have to do with genetic variations within the human population?
Well, okay, so as far as we can tell, there's very little in none of the viral variants, genetic variants that we've seen seem to have any detectable impact on disease severity. That doesn't mean that they can't or that they won't, but none of the ones we have seen so far seem to have any, significantly stronger effects than any of the others. So what accounts for the difference in severity among people, that's still largely unknown. So genetics seem to, account for a fraction, a noticeable fraction, but not even half, not even a quarter of the variation among disease susceptibility or severity of disease. So we still do not know. We suspect that we know that prior conditions, so if you already have asthma or you already have COPD or you already have, some respiratory weakness or disease, then you are more likely to have a severe case of COVID. If you have heart disease or, there's a lot of reasons
Variations
where
in the human population.
Yeah. And so those are the fairly severe ones. If you, for example, have vitamin D deficiency, you're more likely to have a severe case of a variety of infections. So there's things that
with all the sunshine in Miami, Florida, we definitely don't have that.
That's not the reason for the outbreak in Miami. That's for sure.
Sorry, go ahead.
No, go ahead. I'm sorry.
Which is more prevalent, the lipid or the protein membranes in viruses?
That's a good question, and I don't think I can give you a proper answer. There's all kinds of capsids and then and other kinds of structures for viruses, and I don't have the statistics at the top of my head. I can't tell you that.
Okay, so everybody, first of all, getting a question that is for me, not for Dr. Reimers. This immediately becomes a recording. A lot of you are asking if you can see the whole thing. You came in late or you want to share it with other people. If you wait about 5 minutes after I stop the session, it is automatically a recording, and we do have it on the TIES website, tieseducation.org. And so that's the answer to that. Thank you so much. You got a lot of questions there. I think you did a wonderful job explaining everything for everybody. So I want to thank you on behalf of everybody who joined us today. Dr. Reimers, that was really great. I've seen this twice now, and I've learned a lot more even now the second time around. It seems like things are it's crazy right now here where I live. Yeah. So I understand. I want to share with everybody that we have another webinar tonight at 8, and she's actually here with us, Dr. Leslie Jones of Valdosta State University. I put the link right there in the chat, and this is about teaching biology with social justice in mind. Just to give you an idea what it's going to be about, we know that genomic studies have established beyond any doubt that humans do not biologically differ according to the artificial categories that we call races. But despite this, racism is rooted in fallacious, erroneous assumptions of real differences that continue to be destructive misconceptions held by many people. And she wants to share how our science lessons as teachers can assume some social responsibility by providing our students with the current scientific information, demonstrating how race is cultural but not a valid biological concept. So that's tonight. There's your link to join. And thank you a lot for this wonderful presentation. Dr. Reimers has offered to come back and do a few more presentations for us in the fall. So please join my Crowdcast channel. Please follow TIES on Facebook or Twitter and become part of our community. I'll let you have the final word here.
Well, I'm glad to have such interest from so many people, and some people who've come from far away. And please, tell people, spread the word. Science is a way at getting at these really hard questions. Yeah. Hopefully we will, as Bertha said, become less ignorant. So, all right, great. Well,
Thank you, everyone. And thank you, Dr. Reimers. And we'll see you again for another presentation, hopefully in the fall.
great. Okay. Look forward to that. Bye-bye,
Bye,
Bertha.
everyone.
Bye-bye, everyone.